All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. And also with me today is my colleague here at NCCAT, Ed Eng, as well as Lauren Vega from the Pacific Northwest Cryo-EM Center, PNCC, and Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2. This webinar is a regular event. We host the last Thursday of every month, and along with exciting biology, all of the talks we host highlight the methods that research groups are using to obtain and interpret the cryo-EM data they can collect with the help of National Center resources. And we hope these talks are useful, especially for groups just starting to use cryo-EM techniques. Today's speakers, Melanie and Clarissa Durie, were invited by S2C2, and Mike will introduce them properly after my colleagues from each of the centers give a quick update on the cryo-EM resources we make available at no cost to the research community. A quick plug for our February talk. On the 24th, NCCAT will be hosting Liz Kellogg, a professor at Cornell University, who will tell us about her group's work on mechanisms driving molecular innovation, cryo-EM studies of CRISPR-associated transposons. So join us again on February 24th for that talk. And before then, in February, if anyone is planning to attend the Biophysical Society annual meeting in San Francisco, we will have a booth there in the exhibitor hall where you can come talk to some of us in person, but we also plan to schedule some virtual chats for anyone who wants to talk to us but is unable to travel right now. So we're really looking forward to that. Back to today's events, we are recording, and you'll be able to find today's talk along with all of our past talks on our YouTube page or on the events page at cryoemcenters.org. If you have questions during the talk today, please use the Q&A button to send them in at any point. You can also upvote other questions you see there, and Mike will be moderating questions at the end of the talk. And with that, I'm going to turn over to our center representatives. Lauren, I will let you go first.
Awesome. Thank you, Christina. So I am Lauren Vega, and I am the project coordinator for Pacific Northwest Cryo-EM Center. We now offer 2 proposal types. We have a new screening access proposal that we give up to 5 days of sample preparation optimization that we're trying to target towards low resource institutions. Then we have our general access proposal that you can have both single particle analysis and tomography for. For this one, we give up to 480 hours a year, and that proposal type is valid for 2 years. Our general access proposals are also eligible for our training workshop. So at PNCC, we do have a monthly submission deadline for our applications, and each approved proposal, we delegate a SPOC, which we refer to as a scientific point of contact, to ask scientific questions. We currently have 5 scopes. We have 1 Arctica with K3 upgraded to fringe-free, and Arctica with Falcon 3, K3, and fringe-free as well. 2 Krioses with Falcon 3 BioQuantum K3, and 1 Krios with Falcon 4 BioContinuum K3, also fringe-free. For sample preparation, we have a VitraBot, a Leica GP2, and a VitraJet, which is unfortunately still undergoing validation before being offered to users. We can once again have visitors on site starting in March, and we're planning to start our in-person workshops in April, beginning with our sample preparation workshop, followed by our microscope operation workshop in the summer. Look for more information on those workshops on our website and apply today.
Thank you, Lauren. I will turn over to Ed next.
Hi, I'm Ed here in New York. NCCAT is offering 3 main types of access. You can have Krios access. We have 4 dedicated instruments for that. We also have a very robust on-site cross-training program where you can embed here anywhere from 1 week to 3 months. And we have a new grid preparation and screening program where you can have access to our plunge freezers and Glacios instrument. If you want to find out more, you can go to nccat.nyspc.org. Don't forget to come by to our booth at BPS. If you talk to us and find me at our booth, you have a chance to get a chameleon plushie. More information if you follow us at NCCatInfo, which is our Twitter handle. See you there.
And I'll point out, finding it at the booth will be a challenge. So the plushie will be well earned by whoever gets it. All right, Mike, I'll turn over to you.
Hi, I'm Mike Schmid from the Stanford SLAC Cryo-EM Center S2C2. We have 3 Crioses and a Talos Arctica for screening and training. And again, we have all the sorts of equipment that you would expect: Falcon 4 cameras, K3 cameras, and all the freezing apparatus that we offer. And again, here's our website. Please come to the booth. It's right up the street from us, and we'd love to see you in the Biophysical Society meeting. Thank you.
All right, and Mike, I will let you introduce our speakers.
I'm very happy to introduce our 2 speakers today. Dr. Clarissa Durie got her BS and PhD at the University of Alabama and did a postdoc at the University of Michigan, where she and Melanie will be talking about the research that she conducted there. She was the 2020 Bradley Patton Award for excellence in postdoctoral research there, and she has recently moved as an assistant professor at the University of Missouri. Dr. Melanie Ohi received her BS at Pacific Lutheran University in Tacoma, her PhD at Vanderbilt, and she was a Jane Coffin's Child's postdoctoral researcher at Harvard with Tom Waltz, and she is currently the Rowan G. Matthews Collegiate Professor in Life Sciences at University of Michigan, and been elected as a fellow of the AAAS. They are going to speak today about the structure analysis of H. pylori and L. pneumophila type 4 secretion core complexes via Cryo-EM. Thank you. And Melanie will speak first. Welcome.
Yes, thanks for the introduction. Let's see, let's get this started. Start. Okay, yeah, so Clarissa and I are going to tag team today. I'm going to give a little bit of background about how we got into studying these really large dynamic complexes that we're purifying from bacterial pathogens. And then she's going to go into more of the detail about all the tips and tricks that she used to actually push the resolution of the Legionella complex so that we could build molecular models and figure out how this complex fits together. So the bacteria that we're looking at are gram-negative bacteria, and these type of bacteria make up a lot of different types of pathogens that can make you sick. And one of the hallmarks of the gram-negative bacteria is that they have 2 membranes that they have to get things across. So they either have to get the virulence factors out of the bacteria and into the host cell, or they also have to move things from outside of themselves into them. And they have to get it across these 2 lipid bilayers. And so that's a challenge. However, they have come up with many different approaches for doing this. And most of these approaches require these very large dynamic complexes. And so there's a whole bunch of them, T1SS, T3, T4, T6, T7. And the molecular players of these complexes have been characterized using genetics, mass spectrometry, and biochemistry. And there's also many structures of these now too. So for example, the T3SS has some really beautiful structures, also the T1SS. And so we're starting to understand how they fit together. However, the complexes that we are looking at are the T4 type secretion systems. And when we started this project, there actually wasn't very much known at all about what these complexes look like, especially from the bacteria that I'm going to start talking about, H. pylori, which has a very complex T4SS. There has been a number of really beautiful cryo-electron tomography analysis that was done during the process of while we were trying to get high resolution structures. And there were also some crystal structures of what are called minimal type 4 secretion systems that are made of just 3 components. And I just show a few of those examples. And I do really want to give credit to Grant Jensen's lab, Joseph Vogel, Bo Hu, Peter Christie, June Liu, and Craig Roy, because they really did some beautiful work that has helped us actually figure out new ways to purify the complexes and get higher resolution. One of the reasons that we were really interested in the H. pylori T4SS, and this was the first type 4 secretion system that my lab started working on, was because it seemed like it was much more complex than the more minimal system. So rather than having just 3 components, it seemed to, depending on what literature you're reading, it would have 5 to 6 components. And then many of the proteins that were known to be involved in the T4SS, they didn't seem to have natural homologs in the better characterized systems. The other reason that we really wanted to understand how this type 4 secretion system worked is that this is a complex that is required for H. pylori to infect gastric cells and transmit an oncogene called Cag A from the bacteria into gastric cells. And so we started working on this with our long-term collaborator at Vanderbilt, Tim Cover, who is an H. pylori specialist, Borden Lacey, who is also a long-term collaborator. And then we had a team of some amazing scientists, Eddie Chung, who is a postdoctoral fellow in my laboratory, Clarissa, who was also a postdoctoral fellow in my laboratory, Mike Shilo, who was a postdoctoral fellow in Borden's lab, and then a very talented staff scientist in Tim's lab, who was involved in helping us figure out how to purify the complex. And I should say all 3 of the postdocs who worked on these projects have now moved on to start their own lab and are probably really looking for postdocs to join their new labs. Okay, so why H. pylori? So H. pylori is a gram-negative bacteria and infects the stomach. It colonizes 50% of the world's population. And of those 50%, probably 10% will get stomach ulcers. And from those 10%, 3% can get stomach cancer. So that may seem like a small number, but when you have such a large number of people who are actually infected, it turns out that H. pylori is one of the major causes of stomach cancer in the world. And as I mentioned, this is a very long-term collaboration with Tim Cover, who is an H. pylori expert at Vanderbilt. The type 4 secretion system actually moves a small protein, or at least small for EM protein, Cag A, across the 2 lipid bilayers of H. pylori and then into the gastric cell. And this Cag A is a protein that then starts to interact with all sorts of signaling hubs within the cell, and then perturbs a number of pathways, such as the actin trafficking. It also stimulates inflammation and disrupts tight junctions. And so this is a protein that is required for the pathogenesis of H. pylori. And very interestingly, this type 4 secretion system in H. pylori seems to secrete only one protein, Cag A. And there was a model, or at least a cartoon, for how this complex might be assembled. And when we started the project, we were working with a very talented student, Arwen Frick Chang, in Tim's lab, who had an idea of how she wanted to purify this complex. And I only mention this because she was told many times that this was not going to work, but she persisted. So this is the cartoon of Cag A here. And so she decided she was going to try to purify the complex using this chaperone called Cag F that seemed to bind to Cag A, and use this as the handle for pulling down this entire complex. One of the reasons she wanted to do this was because when Tim's lab tried to add tags onto other components of the T4SS, they weren't able to pull out the complex, or sometimes the T4SS didn't function anymore. But CAG-F seemed to be taggable, and so she went for doing this. Now, when she came to talk to me, I told her this was a really bad idea because we were purifying the complex directly from H. pylori. There wasn't very much of it, and then this wasn't actually part of the core complex, and this really was true. So when she looked at the purification, and we can only see it by Western blot analysis, when she ran her purification across sucrose gradient and then blotted for the different components that we knew were going to be involved in the T4SS, you can see CAG-F and CAG-A actually fall off the complex in the gradient. So this should not have worked, but it did. And so she was able to get these complexes into negative stain. And we were able to get enough of these complexes to get some low resolution 2D averages. And if anybody has done negative stain, you might notice that, I'm not showing really large fields of view of these complexes. And that's because even in negative stain, it was very hard to find intact complexes. But we could see it was very beautiful structure that had some symmetry, and here was a side view. And then she was able to make some deletion mutants that allowed us to at least map where we thought different components of the complex were. And then we were stuck. So this was 2016. We were stuck for a very long time trying to figure out how to get more of this complex and get it stabilized so that we could get it into vitrified ice and actually get a 3D structure. And while we were working on this, there were new structures coming out using cryo-electron tomography. And this actually did give us some hope, because here's an image of our 2D negative stain 2D average. And then here is a cut through of a tomogram from Grant Jensen's lab of the H. pylori Cag T4SS. And so it did look like we were actually getting a structure that was holding together. And so we were probably on the right path, at least. So when I moved to the University of Michigan, Eddie joined the lab, and working with Nihaj, they started working on trying to really improve the purification. And the challenges that they were dealing with were low concentration, preferred orientation in the vitrified ice layer, severe aggregation once we could even see it in vitrified ice. And so they worked on screening various detergents, buffer conditions, and different types of cryo-EM grids. And finally, we're able to get this complex into vitrified ice in a way that we could get images and get 2D averages. However, this was still a really challenging project, because each image had between probably 1 and 5 complexes, and sometimes 0. And so there was a lot of EM time to get enough particles. Eddie persisted though, and we were able to get this beautiful structure. And this actually was the first high resolution structure that we got at the University of Michigan. And it was super exciting. So you can see we had, there's this cap region, this mushroom cap region that we call the outer membrane cap. There's a periplasmic ring, and then there's this kind of ugly stalk. And this is because hardly any of the complexes had this stalk region. And so we really don't know what symmetry this is or what it's composed of at all still. And I just want to do a cut through of this. So this would be where the outer membrane sits, and this would be where the inner membrane sits. And you can see that there probably is some channel that goes through the complex. And I think the other thing that you can see here, and this is a composite structure because we have applied different symmetries, so there's no connection between the periplasmic ring and the OMC. But you can see that there is a lot of molecular detail within the center of the complex. Now, there were really no crystal structures of any of the components of this complex. And that, I think, is because when we started to really look at it, all the different components were really interdigitated. So if you were trying to purify individual components, they just didn't fold. And what we noticed right away was that this OMC had a 14-fold symmetry, and then this periplasmic ring structure had a different symmetry, a 17-fold symmetry. And Eddie worked very hard to get the resolution to a little bit under 3.5 angstrom resolution. But with the complexity of the map and the lack of any sort of crystal detail, we needed a lot of help to build the models. And this is where Mike Shelow entered the picture, who did an amazing job building models from these very challenging maps. And I'm not going to go into huge detail about this because it's published and probably people don't care that much about the details. However, these are going to be themes that I think Clarissa will probably talk about in her talk, is that what we found is that these large complex T4SSs actually, they use their building blocks in many different ways. And so the proteins are not in a 1-to-1 to 1-to-1 ratio. We had, one CAG Y, one CAG X, 2 CAG Ts, 2 CAG Ms, and 5 CAG spores in each of the asymmetric units. And really interestingly, depending on where they were in the complex, some of these proteins actually, even though they were the same peptide, have different structures. And so that was a surprise. And then the other really big surprise was that the periplasmic ring had 2 of the same components that were actually found in the OMC. So these components were actually bridging the asymmetric unit. And that was a surprise. We really thought that, that those were going to be two different peptides, but they were not. Now with H. pylori, we were really struggling because we just could not get enough materials and to actually see a higher resolution structure without symmetry imposed. So this is a very low resolution structure. And this is just to show you that we can see how the periplasmic ring is actually connected to the OMC, but not with any sort of detail that would allow us to actually identify how or what molecular components are doing that. And then we could also see different connections that were not, we're not going across this symmetry mismatch. The other thing that we did not see was the CAG A, the protein that gets translocated. But there is a crystal structure of part of CAG A. The other part is predicted not to be folded. And just to show you the size comparison of this, so CAG A could actually fit within the periplasmic ring without unfolding. But if it needs to get across the outer membrane, either there's going to need to be a huge conformational change in that OMC cap, or CAG A is going to have to be unfolded going through the complex. And this is something that we're still trying to figure out, but I just wanted to show the size comparison. The other thing that we could look at was how different is this large complex T4SS compared to the structures of the more minimal complexes. And we had hypothesized that maybe the Cag T4SS, because it only secretes one protein, might have some sort of different type of channel that goes through the outer membrane, or at least a different size. And that did not seem to be the case. And then the other thing that we were able to figure out with our model is that even though there was not sequence homology between many of the more complex T4SS components, there was a high degree of structural homology. And so they seem to be organizing at least in the core region, in a very similar manner of the more minimal systems. And then they just add on to these other components, making the complex larger. And so this really led us to have some questions. And we had a structure, but we really didn't understand how the symmetry mismatch was accommodated. We were wondering if we could visualize different conformations, because one hypothesis is that maybe that the region that goes through the outer membrane might, have some different conformations. And then we really still have no clue about how there are differences in substrate selectivity. However, this system was really hard to work with. And so it was so hard to get enough complexes to actually get better structures. And so the practical consideration was, can we work with a system that's actually easier to purify? And this has led us to move into Legionella, and I'm going to let that will be the next talk. And so Clarissa will take this, but like Legionella also has one of these really large complex T4SSs, but from the tomography, we knew that there seemed to be a lot more of them within the bacteria. And so we thought, well, if we could get that purification to work, maybe we could get more complex in vitrified ice so that we could get higher resolution. And also we could then directly compare two T4SSs that had been really predicted to have very similar structures. And I'm not going to go into huge detail on this, but just to say that once again, this is a collaboration with Mike Sheedlow, Borden Lacey, and then a Legionella specialist here at University of Michigan, Michelle Swanson, who helped us figure out how to grow the bugs. And then very quickly, Clarissa was able to figure out how to purify the complex. This is a negative stain. And then she did mass spectrometry to show that we were getting the components that we were expecting. And then I'm going to let her talk about how she got it into vitrified ice. I'm not going to go through everyone who is in my lab. I think I've mentioned the people who have worked on this, but I do want to give a shout out to both the national centers. We collected some of this data at the Pacific Northwest Center too. And then we have a really great EM facility here at Michigan who helped us do some of the initial work. And then I would also like to say that we're looking for postdoctoral fellows in the OI lab to work on this and other projects. So I will turn it over to Clarissa.
All right, and I'm gonna share my screen really quickly. So to sort of pick up where Mel left off, as she said, we were able to purify these complexes, and building off a lot of what Eddie had already learned, we moved really quickly into vitreous ice, which is what I'm showing you here. So this is from our 2020 publication, and this is what I think of as the side view of the 3D reconstruction of this complex. And I've just placed it over kind of a cartoon diagram of where the membrane would be. And you can see over on the right-hand side, this is another example of that great Cryo-ET work. This is from Jun Liu and Craig Roy's group, showing again that our structure looks like what is in the intact structure. So that was really promising. And then just again to review the terms, sort of the parts of this that I'll talk about as I dig into a little bit more about the data processing details. We have the outer membrane cap, which is made up of a flat disc and then a dome that sits on top of it, and then the periplasmic ring. So this is again very similar to what we saw with the H. pylori complex, but I'll talk about some differences as we go. So I'll use the full terms when I'm talking about these regions, but you might see PR and OMC in some of the figures that we look at. So if we turn that view towards us 90 degrees, now it's as though we're outside the cell looking in. This is what I think of as the top view of the complex. And we can see those really striking 13 arms that extend out radially. And this was expected from our negative stain work, and again, earlier work by other groups and tomography and other examples. And you can also see that the density is not really even, right? We're missing some information about the very center of the complex. And so Eddie and I sat back to back in the lab. And so this was really interesting because that part of his complexes, of the H. pylori Cag complex, always looked really beautiful. So it was kind of strange that this part looked a little bit more disordered. If we flip it over 180 degrees, now it's as though we're inside the cell looking out, and we're looking at the periplasmic ring. And so here, just to make it a little bit easier to see, I'm highlighting each of those repeating units, and there are 18. So here, the degree of symmetry is different from what we saw in the Cag complex, where we had 14 and 17-fold symmetry, but the symmetry mismatch itself is conserved. And we actually found that that's not just the case between these two type 4 secretion systems, but symmetry mismatch between different regions is something that's seen in type 2 and type 3 and type 6 secretion systems too. So sort of is emerging as potentially an important feature in and of itself. So the global resolution for this complex is about 4.6 angstroms, which was very exciting at the time. This was the highest resolution structure that we had for the Legionella type 4 secretion system core complex. But Mel mentioned there's not a whole lot of homology, right? There was not a lot of sequence similarity between the components of the Legionella system and the components of the other systems that had been studied. So what resolution do we really need? If we had models, if we had crystal structures or good, homology models, then 4.6 angstroms might be good enough to really figure some stuff out. But because that wasn't the case, we really needed to push to higher resolution in order to be able to figure out what exactly was going on in this complex. And luckily, we were able to use that symmetry to our benefit. And the way we did that, there's a sort of an overview of the data processes over here on the right. But the big takeaway is we were able to treat each of these highly symmetric regions separately. So I did particle subtraction to do focused refinements on one area of the complex at a time. And then we were able to use that symmetry and basically increase our signal to noise and really improve the resolution to the range where we could now start to build in models for the protein chains, or at least polyalanine models in different cases. In doing that, we do, of course, lose some information, though. So as Mel had mentioned, we took a similar approach with H. pylori, and when we do these focused refinements, we lose information about the connection between these two mismatched regions. And then the other thing that I noticed was in, applying this strategy, the dome area never looked any better. It kind of would just get smeared out. And so it sort of suggested that this may not have the same symmetry as the rest of the outer membrane cap in the case of Legionella pneumophila, which again was different from what we had seen with other well-studied systems and kind of just remained a big question mark. But we were able, as I mentioned, to build in a lot of models. And this again is back to Mike Schiedlo. Mike built all of the models, all of the protein models that I'll show you throughout this talk. And this is, I'm just gonna hit on a couple of my very favorite findings so that we can move through relatively quickly. But so this is the asymmetric unit of the outer membrane cap disc. So there would be 13 of these forming that ring. And in this image, each protein is shown in a different color. This is the same asymmetric unit, it's just tilted 90 degrees, so you can kind of see all of the different parts and again, how closely they're all fit together. If you think more spatially, on the left-hand side, this is the electron density map color-coded in the same way. So this asymmetric unit would correspond to like this portion of the map. And we learned a ton. One of the surprising things was that we saw 2 copies of .D in the asymmetric unit. Now we were less surprised because we had the benefit of the work on the CAG system where we saw all of those different stoichiometries, but we hadn't been anticipating seeing 2 copies of this protein. And then we were able to identify some of the predicted core components, .C and .H. So again, a lot of biochemical and genetic experiments had gone into identifying the 27 components of this complex and the 5 components that were known to make up the core of the complex. But then we also identified 2 more proteins. So .K was one of the proteins that we observed. It's shown here in this kind of light blue. So this has a dot name. It stands for defect in organelle transport. That's one of the ways that the complex was named for the function that it has in the cells. So that was a known component, but it wasn't known to be part of this core complex. And then LPG 0657. So this is a gene number rather than a dot name or an ICM name. At the time we found this, there's one paper from 2016 from the Nino lab that showed that it was an important protein for efficient intracellular replication. But again, it wasn't really known to be part of the complex. So that was really exciting and intriguing. But you'll also notice there are a few gray chains within the structure here, and those are proteins that we weren't able to identify yet. So still some open questions. In addition to those 3 unidentified chains in the outer membrane cap, we also had 2 polyalanine chains that we were able to build into the periplasmic ring, but we weren't able to identify what those components were. Again, we didn't understand how the symmetry mismatch was accommodated between these regions and had a big question about what was going on with the dome portion of this map. And then we had to undergo some renovations at the University of Michigan. All of our microscopes were down, and we were very fortunate to be able to collect more data at the Stanford Center at S2C2. And so what you see here, the 2020 publication was collected at Michigan on our G2 Cryos with our K2 camera. And again, I am very excited and proud of this work, but you can see, so this data set actually was collected over 5 days. In just 2 days at S2C2, pardon me, I think it was 3 days, less time. We got way more images, which meant a lot of particles. And then of course the K3 also has the energy filter, so really high quality data. And so now what you'll see in this video is a comparison of the maps that we had from these two. So right now we're looking at the 2020 publication. This is the C1 map, so we haven't imposed any symmetry yet. And again, you can start to see that there's some connections between these two regions. We see broadly these different areas, but we're not seeing any detail really in these arms. We're seeing that incomplete density in the dome. And then now in the darker gray, this was again a C1 reconstruction, but with the bigger, better data set that we were able to collect on the Krios G4I with the K3 camera and the bioquantum energy filter. And so you can see there's just a lot more detail. So this was already, really promising. But we still, wanted to push the resolution as far as we could to make sure we could identify as many of these components as possible. So we followed a very similar workflow in terms of, doing focused refinements and really capitalizing on the high degree of symmetry in the different regions of the map. And as you can see, we were able to improve the resolution substantially to below 3 angstroms for the refinements where we were able to impose symmetry. So we're about to get an extreme close up here, just so you can kind of appreciate the quality of the maps, just these beautiful side chain densities that we were really excited to get to work with. So we were able to identify a lot of those components. So I'll mention LPG0657 is one of the proteins that we identified previously. We actually were able to extend those models with these helices that extend, kind of upward from the flat disc. We also realized we gotta have some better names here, but we were out of both DOT and ICM names. They're all named as DOT A through DOT Y right now, I think, but we had identified 3 more proteins. So we decided to call them DIS for defect in organelle DOT for DOT ICM secretion. So we have DIS1 here. We also identified LPG0823, which we call DIS2. And then that really striking radial arm is LPG2847, which we call DIS3. So if we look a little bit at DIS2, this is really interesting to me because it's right there in the pore where you would expect proteins to need to pass through. And it's relatively small. It's got 2 domains. They're very similar, and they're actually held together by 5 cysteine bonds, which was, again, just kind of surprising. We didn't know anything about this going in. It's not anything that had been predicted to be part of the complex. And then if we look at DIS3, LPG2847, this is an example showing the maps at different levels within this part of the structure. So the top, the C terminus would be near the interior, near the center of the structure. And we had the best resolution there. And you can see it gets a little worse as we go further out. But if we look at rung 13 here, this is one of the things that was really helpful. Mike was really excited to see. You can see there are 3 tyrosines right next to each other. So in building in, residue by residue, this was a really important, feature to be able to figure out which protein was actually making up this component. It was also, really interesting to put this back in the context of that tomography work that had been done previously and see how complementary they really are. So if we place one asymmetric unit within the map from the tomogram, you can see that it fits really nicely. And we also see that those alpha helices from DIS1 tend to point straight up and actually fit into some additional density that was observed in the tomography work to connect to that, outer leaflet of the membrane. So the other thing that was interesting when we think about the proximity of our complex to the membrane is that we were able to identify 5 additional likely lipidation sites. So if you put it all together and you think about the fact that there are 13 copies of this asymmetric unit around the ring with 5 lipidation sites, 2 helices that extend up possibly into the membrane, and then actually the surface of DIS3 is positively charged. So you can imagine, favorable interactions with the phospholipid head groups of the membrane. It suggests that this part of the complex is really well anchored into that membrane layer. Now I want to move briefly to the periplasmic ring. I'm not going to ask you to remember any of this, but I'm just going to show you that we were able to identify these proteins as .F, .G, and .H, which was great. We knew we were, missing some of the known core complex components. now we can see where they are and how they fit in. But you'll notice here we have .F, .G, and .H. If we go back to the components of the outer membrane cap, we also see .F in the outer membrane cap. And so you might be thinking, aha, this must be how the symmetry mismatch is bridged. But in fact, that's not the case. What we see is that it's the same domain of .F that's present in the periplasmic ring that's present in the outer membrane cap, which means there are 18 copies of this protein in the periplasmic ring plus 13 copies in the outer membrane cap, which gives us 31 copies of this protein total, which again was just a really unexpected stoichiometry. But it didn't tell us how that symmetry mismatch is bridged. And so now I just kind of want to highlight the point that really having this really large data set, having more particles is one of the main things that gave us enough power to figure out what was going on there. So what you're going to see in this video, this is the C1 reconstruction. So this is before we did any particle subtraction. There's no symmetry imposed. And everything is shown in gray except for one component, which is .H. So now we're as though we're outside the cell looking in, and we're going to kind of slice through. Now we're in the plane of the outer membrane cap disc, and you can see there are 13 copies of .H. That's actually the C terminus. And then as we keep going further through the complex, we get to that space sort of between the outer membrane cap and the periplasmic ring. And we see these 5 lobes of density. Of course, 13 plus 5 is 18. So as we go further through the complex, now we see all 18 copies of the N terminus of .H in the periplasmic ring. So if you see still images better, this is the same information in a different representation, just showing that it's in fact .H that's bridging that mismatch and symmetry, right? We have all 18 copies in the periplasmic ring. 3 of them extend all the way up into outer membrane cap, and then 5 are sort of in that in-between space. But we were still really hung up, or I was still really hung up on what was going on with this dome portion. This is the part that should form a pore, and it's so clear and beautiful to see in the other structures that had been studied. But there was a new tool available right about this time. CryoSpark came out with their 3D variability analysis, which allows you to, as it says here, explore heterogeneity in your data set. And so we were able to see that, doing this type of principal component analysis, we were able to see that there is a distribution of conformations where the dome is in a different orientation relative to the rest of the complex, or relative to that outer membrane cap disc. And so in order to do this type of analysis, I had to downsample the particles, right? These are really big box sizes, and running the jobs on GPUs, they just weren't able to run the particles at the approximately 1 angstrom per pixel. And so I had to downsample the particles, which means we ended up with a lower resolution. We're at about 4.6 angstroms. But I could finally see what was happening in the dome, which is that it has 16-fold symmetry. So that's yet another symmetry mismatch between the dome and the disc now, as well as between the OMC disc and the periplasmic ring. The other thing, if you remember, this is the range where I said if we had a good model, we could start to fit it into the map. And I also made it kind of a big deal of saying there's no sequence homology. There are no good models. This is the one exception. So the C terminus of .G has a really high similarity to the archetypal system Verb-10 and Cag Y in the Helicobacter pylori complex. And so we actually had built this model. Mike built this model in our 2020 publication, but we found that it fit beautifully into the density that we were able to get out of that 3D variability analysis. If we impose that 16-fold symmetry on our model, it again fits really nicely into the map. So that was really exciting. The other thing that I'll show you that's a little bit speculative, again, I want to point out that this is an animation and not a movie. Of course, we don't see motion in our single particle frozen samples. But when we put all of the details of the model into what we saw from the 3D variability analysis, all of those different conformations, they appeared to be distributed continuously, right? They're not totally separate sets. And so that suggests that there could be motion that's happening, that's leading to these different conformations that we observed. And so that's what we're showing could possibly be happening here. And I also will remind you that we have good reason to think that this outer membrane cap disc, the flat part, is really well anchored into the membrane. And then you'll see it looks as though the dome and the periplasmic ring could have motion relative to the disc that's so well anchored into the membrane. So of course, we would want more experiments to figure out what's going on there and see if we could confirm this idea. But I think it's really interesting in the context of, again, the fact that we see so many symmetry mismatches in different types of secretion systems. So this is just an overview side by side of sort of the workflow for these two papers that were very close together in time. And again, one of the biggest differences was the amount and the quality of the data. And that was due in large part to the facilities that were available at S2C2, although I should say we now have similar instruments available at Michigan as well. But having access to the national centers was a game changer for this project. And then the constant advancement in the software and analysis options that we have available to us, the 3D variability analysis tool made a big difference in this project as well. But there's still a lot that we don't know, right? All good science leads to more questions. So we've identified core components. We don't know yet if they're essential for pathogenesis, and we don't know if we've identified everything. There could still be others that we haven't identified yet. We talked a little bit about the symmetry mismatch, the conformational heterogeneity, and what some possible functional implications are for that. And then something to maybe emphasize a little bit more is the difference in substrate repertoire between these type 4 secretion systems. So Mel told us about how the CAG system has this one substrate protein, CAG A. The Legionella system is thought to have as many as 300. So I'm really curious to know why they work so differently. And one of the things, when the dome region was very elusive and we weren't sure what was going on there, is that we thought maybe the dome is much more flexible. Maybe it's just wide open. It's just letting everything through. But it turns out it's actually very similar in size and shape to all of the other well-characterized systems. So we can rule that out, but there are a lot of other possibilities that still exist. And I have a long-standing interest in protein translocation. So if you, the teal structure here is basically everything that we've shown you so far today, but then you can see that's not the whole complex, right? In the CAG system, we could see sort of a low resolution stalk. We haven't yet seen that in our single particle data from our isolated complexes from Legionella. So I'm really curious to understand what's happening on this side of the complex. And I have the opportunity to do that now in my lab at Missouri. So we'll be digging into the mechanism of translocation and the effects of the cell life cycle on that process. My lab is now open. I just started, like I said, University of Missouri. I'm in the Department of Biochemistry. If you're interested in talking about the work that we do and seeing if it might be a good fit for you, you're welcome to email me. Potential graduate students can also learn more about the program at the link here. And I also should mention that Missouri has invested heavily into a really great imaging facility that has a lot of great biological specimen equipment, including a Cryo-EM and an Aqualos, and then also a lot of really great instrumentation on the materials science as well. So I will take just a little bit longer to dig into the acknowledgements a little bit. So all of this work was done at the University of Michigan. Mel is an amazing advisor. I hope you consider coming to join my lab, but I also can't speak highly enough about the work that I was able to do and the mentorship that Mel has provided for me and for the other folks in the lab who, as she mentioned, have gone on to do the things that we want to try to do. So again, I worked closely with Eddie on a lot of this. Eddie Chung, he's now a PI at the Catholic University of South Korea. And of course, Mike Schiedlo, who's now an assistant professor at the University of Minnesota. Michelle Swanson's group, in addition, has been really great, as Mel mentioned. My work was funded in large part by the NIH, by an F-32. And then last but not least, I'll also mention the Leading Edge, which is a group for women and non-binary postdocs who are aspiring to faculty positions. And it's a group that I was able to join and kind of get a lot of help on the academic job market and support in that regard. And of course, now as we all kind of go through the process, it's also a great resource for new assistant professors. So applications are open for that program until February 4th. And I'm happy to chat with anybody if you have questions about that too. But that is it for me. I think now we can take questions.
Thank you, Clarissa and Melanie. Melanie has been answering some of the questions as they've been proposed. Melanie, would you like to clarify or share your answers for some of those questions? Uh, and more questions are available if you just type them into the Q&A, please. Thank you.
Okay, so one question that I think that is worth addressing, especially in this kind of forum, is how do you build models into maps that maybe aren't so great, like between the 3.5 to 3 angstrom level? How are you sure about that? And I think that's something that the EM field is really grappling with right now. There are more and more tools that are available. We didn't feel confident enough to do that in this system, which is why we started collaborating with Borden and Mike. And I think we take a more conservative approach. So when you can see the side chains and do that, we could see the side chains. And so we're able to identify the proteins. But you do have to be careful, because at one point in the initial structure that Clarissa got, we could not figure out what those arms were. And we had a pretty good idea what it was, we thought. We built that model, it fit. And then in the end, we looked at a mutant and we deleted that protein that we thought it was, and the arms were still there. So, you have to be careful, because you can really get it wrong. And I think between all of us, we spent a lot of time looking at the maps. And I mean, of course, Mike the most, but there was a lot of checking back and forth to make sure that we weren't going down a rabbit hole, because you can get really excited. So I did want to address that question, because that's, I think, something that, like I said, we're still kind of grappling with as a field.
Okay, and we have another one. Our predicted models, that is AlphaFold, RosettaFold, the current hot things, are they useful here, especially considering how tightly the interconnected the subunits appear to be, and if that in fact affects the problem that people have seen with AlphaFold is that yes, it predicts individual proteins very well, but if they happen to be very much involved in oligomer associations, that throws AlphaFold for a loop in some places, or in a
Yeah. So yeah, I think for AlphaFold, this all happened before AlphaFold, so that was the first answer. It might have worked for the .G, right? Don't you think, Clarissa and Mike? I know Mike can't answer, but I think .G, it might have worked. I think it might work for the stocks, actually, because my guess is that might just be 1 or 2 proteins that are kind of stretched out. But for many of the others, I don't think it would work, because we also had multiple copies that had different structures depending on where they were in the map. And so luckily for us, AlphaFold won't make us obsolete, at least this lab yet. So.
I had a question. So the symmetry mismatch is very, very interesting. I mean, and I thought that symmetry mismatch often is a harbinger of rotation and shifting relationships between sets of subunits, and the symmetry mismatch just allows that flexibility, because you don't have fixed relationships there or they shift. What I did, it was very interesting, was that additional 5 units that were more well resolved than the others, and the spacing between them, spaced 4 between, and then 3 between, then 4 between, then 3 between. Is that something we can take from how symmetry mismatch is expressed, and what it might be, what's it good for?
So the way I kind of think about it, at least in the Legionella structure, there's a little bit more detail that I didn't have time to go into, but we can actually see the subunits that have the lobe that does not cross the symmetry mismatch. So they're kind of in that in-between asymmetrical plane. They have varying densities. Some of them are easier to see than others. And when you look at individually, like Mel had said, some of these proteins have slightly different shapes. The amount of a curve or a kink that's between the part that's in the periplasmic ring and the part that's not varies. And so I have wondered if it might be the case that those subunits can slip in and out, and maybe they take turns being associated, being integrated into the outer membrane cap, or being sort of in between that space. So I wonder if that might be one of the ways that symmetry mismatch is translated into motion or flexibility.
Okay. We have another
I'm
question.
going to chime in just for a minute. Some of you may have to run off for another meeting at 1. So I just wanted to, while everyone is still here, thank Clarissa and Melanie for that beautiful talk and making it all look much easier than we all know it was. That's quite a lot of work. And thank you for joining us. Please join us again next month if you want to get into some more nuts and bolts. And come find us at Biophysics if you're going to be there. And we've got a couple more minutes for last questions.
Good. Keep typing them into the Q&A. We have time for a few more. One question. Do you believe there is space for the extracellular pilus to begin from the inside of the outer membrane complex? So this is probably what's sticking out of the cell. In the Legionella, is it possible that the intermembrane complex moves together or in conjunction with the VIRB10 like outer membrane helices?
So that's a really good question. And so I will say a lot of type 4 secretion systems have a pilus. The Legionella type 4 secretion system does not. Mm-hmm. But yeah, again, from a variety of different sort of inputs. So like even biochemical assays, sort of differential centrifugation assays have shown that .G is expected to associate with both the inner membrane and the outer membrane. So we know that. We also know now from our structures that .G is the part that makes up the outer membrane cap dome, the part that makes that pore. I didn't mention it, but it is also one of the components that we see in the periplasmic ring. And then, like I said, there's this biochemical evidence that it also associates with the inner membrane. So it's kind of thought that .G might sort of be the center of the whole complex that everything else is sort of built onto. Now, what I think it's really interesting is that we see it in different copy numbers too, though. We see it 18-fold in the in the periplasmic ring, and then 16-fold in the pore. And so we don't know what it might look like in the stalk region or the inner membrane complex or wherever else it might be on that side. But I do think that it's going to be important in, sort of coordinating whatever's happening from one end of the complex to the other.
Mm-hmm. Yeah, and the idea that you have the same protein associated with both the outer membrane and the inner membrane, those that's generally not the case. That you have stuff that's specific for the outer one, and specific for the inner one, or specific for the periplasmic, which is different from both. But to see the same protein participating in all of those, that's very, very interesting. Mm-hmm. In different copies. Let's see.
Can I ask one more question, which actually is a follow-up to one of the questions in the Q&A? Someone asked about tricks for choosing grids and detergents, and Melanie answered it's trial and error. But I'm wondering, in this particular case, how many trials were you willing and able to do?
Eddie did a lot of trials. I did not do so many.
And then years before that, so 5 years of trials. Yep.
Yeah, and I do think it's something, I mean, when we think about, getting a more stable complex, I do think it's something that I would like to try some more different things, more on the purification side than maybe on the, like plunging side. But Legionella are kind of finicky to grow. So whenever we, had a prep, I was also a little bit protective of it in terms of, trying a lot of different things.
And again, another question about symmetry mismatch, that, okay, you have these different symmetries in the different rings, but apparently in some cases there's an extra adapter protein in between that perhaps is not well defined, that's only in the symmetry mismatch area. Which, how would you approach something that you think might be just in the symmetry mismatch part of the complex?
You have to get a much higher resolution at C1. So biochemistry. And it's very possible that we're missing components. That would be great, actually, because that would continue the project. But you can't apply symmetry, you'll lose it.
Yeah, right.
So you need to get more particles.
And align them so that every symmetry mismatch acts the same way.
Exactly. Yeah.
Yeah. Where do you start? Which is subunit number 1 with respect to all the rest of them? That's an interesting problem. Okay.
All right. Well, I think we won't quite have time to get to the rest of the questions, but I think we got to some really interesting ones that cover quite a bit of the workflow. And unless you guys had any last things you wanted to add in, we'll wrap up for today.
Great. Thanks for having us.
Thank you very much.